Did you know we had two unique candidates coming in today for the interview position of Biology 111 department at LMU for the 2019-2020 year?
Yeah, I heard they range from 1 millimeter to 30 meters. Look.
Pretty flat.
They're covered in small microscopic hairs, and it's kind of gross, but they produce mucus to travel. But what's cool is that they have light detecting sensors instead of eyes. I hope they live up to our standards because it's very hard to pass our interviews.
No way. I heard that they have bilateral symmetry.
What?
Yeah, I know. What's crazy is that they have no head and no more than 2 layers of cells and tissues and organs. I also heard that they contain not one but 2 openings for digestion, a mouth and an anus. They even do respiration through the skin.
Well, they seem promising. They even have a functioning cardiovascular and nervous system. What's not to like? Even though they're acoelomate, they can reproduce sexually and asexually.
Wow.
What did you say their name was again?
Their names are Nemertea. Oh look, there they are. Are you guys here for the interview at the biology department at LMU?
Hi, yes, we are Nemertea.
Hi, nice to meet you guys. So tell us a bit about yourselves.
I'm Nemertea, but you can call me ribbon worm. I've lived in the ocean my entire life, but I have a couple of relatives that live in fresh waters. I even have a couple of distant relatives living in terrestrial areas.
Well, I have sexual dimorphism. Although my other family who lives in freshwater is hermaphroditic, our eggs and sperm are released into the water and we fertilize externally. Reproduction is by asexual fragmentation or sexual, when it is normally gonochoristic. We are very complex.
Well, do you have any preferences on food? We have a strict no egg diet.
I mostly eat on small invertebrates, like crustaceans and annelids, my favorite if I may add. But Tyler likes to eat on eggs and other invertebrates. If I'm trying to have a 5-course meal, I'll occasionally eat microbes that have, of course, been filtered out beforehand.
Interesting. I'll take that into consideration. On to the next subject. How will you get here? LA traffic can be pretty time consuming and we need workers here on time and ready to go.
Well, my locomotion is through highly developed muscles that allow me to contract my body by shrinking to a 1/10 and extend my length when I feel threatened. I live right over by Seaver, so I am confident that I can make it to LSB for work on time.
Well, I could probably make it to U-Haul in that time, but I guess I don't have any competition anyways. I have a unique, aversible proboscis lying dorsal to the gut, which is how I catch prey. I can glide over the surfaces by ciliary action aided by muscle secretions. I have appendages and a hydrostatic skeleton.
I am an unsegmented phylum,
Me too.
but we have no development patterns.
Well, they seem like amazing candidates.
My concern is that if my finger touches them, it'll swell up. I'll have to wear gloves, which is really inconvenient.
I did a bit of a background check on our 2 candidates to make sure that they were genetically qualified. Look at this phylogenetic tree I made.
No way, I made one too. But I did one with data from their characteristics. Tell me about yours.
A phylogenetic tree is a branching diagram showing the evolutionary relationships among various biological species. I made one with data from their DNA. The further down you go, every node is the most recent common ancestor of all species in those branches when regarding DNA. All the species shown are the present-day species. Porifera has the greatest amount of similarities in its DNA to the species' original ancestor because it branches off from the original node. The next node is the next most common ancestor, and the next node branches off to Cnidaria and multiple other species. Therefore, Cnidaria is the most related to that specific ancestor. Also, the DNA of Echinodermata and Chordata are the most similar because they share a common ancestor. Annelida has the same common ancestor that includes Arthropoda, Nemertea, and Mollusca, but Nemertea and Mollusca have the most recent common ancestor that doesn't include the rest of the species.
Cool. Well, my phylogenetic tree is made out of the similar characteristics. The further you go down the tree, every new node shows the most recent common ancestor of all the species that share the most common characteristics. All the species shown are the present-day species. Porifera is the outgroup and has the most similar characteristics to all of the species because it branches off of the original node. The next node branches off into Annelida, who's the next common ancestor, and that branches off into Nemertea and Nemertea.
It seems like ours are pretty similar, because Porifera is the most commonly related species in both phylogenetic trees.
Very true. It seems they are different because they have different common species, because one is based on DNA and one is based on characteristics. Although they look pretty similar, they aren't. Our results of comparing and contrasting these trees mean that just looking at the characteristics isn't precise. Looking at the DNA is more accurate, and also characteristics can arise through convergent evolution, so looking at DNA is always better.
Leah, do you know of any pre-published phylogenetic trees that we can relate to the ones that we made?
Yes, I actually found one. It explains the evolutionary history based on the genetic sequencing. The reason why this tree is different from ours is because it has a wider variety of organisms. The closest relative to Nemertea is mollusca.
Well, based on their interviews and their phylogenetic trees, I say they're both hired.
I couldn't agree more. We'd like to thank TA Aaron, Dr. Olet, and the biology department.